Hi, my name is Arsheya Sharma, and I'm currently presenting from Washington, US. My mentor Lauren and I worked together to write a review paper on for one of the changes and one of the three organisms for the other change. So first off, I will discuss the changes in locomotion using the examples of Toxoplasma gondii and Spinochordotus tellini. Um, Sorry, I didn't realize I was unmuted. So I'll be focusing on the changes of um, in locomotion using the examples of Toxoplasma gondii and Spinochordotus tellini. These locomotive changes cause the host to seek specific predators and perform self-sacrificial behaviors, respectively. Then we will move on to the example of Dinocampus coccinelli to talk about the changes in nutrition and protection provided by the host to the offspring. Uh, So Toxoplasma gondii, what does it do? It alters behaviors towards cat urine and mice, and it alters the rodent's perception of cat urine, which is usually avoided because of the ecological relationship between cat and rodents. It's a predator versus prey, but with the presence of Toxoplasma gondii in the rodent system, it causes an attraction to the cat urine, which is normally avoided. It does this by modifying hormonal and neurotransmitter communication, and it directly interferes with neurons in brain areas controlling behavior, which causes the mice to show attraction to cat urine and thus changing their predation-related activity. Another crucial mechanism that Toxoplasma gondii uses to control over host behavior is neuromodulation, and it does this by blocking the anxiogenic and methyl diaspartic acid receptors in the amygdala, a region that is associated with fear processing, or administering serotonin antagonists, which produces a behavioral change in rats that mirrors the fiercelessness displayed by T. gondii infected rodents when approaching cat odors. So such findings suggests that the parasite's influence might involve molecular pathways associated with anxiety regulation. Moreover, the alterations in neurotransmitters levels adds another layer to this dynamic T. gondii infected mice, demonstrated elevated to this dynamic. So T. gondii infected mice demonstrate elevated levels of substances like dopamine, norepinephrine, and homovanillic acid, which are molecules that are integral to mood regulation, memory, learning, and even cerebral blood flow. And thus, by manipulating these neurotransmitter levels, T. gondii could potentially impact the fundamental drivers of rodent behavior, which contributes to their altered stance towards cat urine. The next example, Spinochordotus tellini, it is a parasitic hairworm that infects Mechanoma thalassium, which is a type of grasshopper. And these infected grasshoppers exhibit abnormal behavior, like jumping into water, which helps the hairworm complete its reproductive cycle because it needs an aquatic environment to reproduce. But this causes the grasshopper to drown as it can't swim, and it releases the parasites into their ideal reproductive environment. So some changes that were observed in grasshoppers, infected grasshoppers, were molecular changes in the CNS, which is the central nervous system. And these findings revealed that specific protein families involved in the CNS development exhibited selective expression in the manipulated grasshopper, which suggests that the grasshopper's brain tries to defend against the parasite's attack. And after recognizing the intrusion and attempting to safeguard its neural integrity. Furthermore, this study pinpointed proteins in the hairworm that were associated with neurotransmitter release and apoptosis regulation during the manipulative process, which indicates that the hairworm strategically influences chemical signaling inside the grasshopper's brain, potentially inducing apoptosis in the CNS to further exert control within the grasshopper's brain. These revelations align with previous research on other host parasite systems, that illustrating that the widespread phenomenon of parasitic manipulation of host behaviors through cellular interactions affects critical brain processes and neurotransmitter activity. And using this, parasites can steer host behavior towards actions that serve their own reproductive goals. The next one will be a change in how parasites cause hosts to provide protection and nutrition to their offspring. The example for this is Dinocampus coccinelli. It is a ladybug infected with Dinocampus coccinelli exhibits a behavior known as bodyguard behavior, which is when a parasite causes the host to show hostility towards anything that might be a threat to the maturing larvae injected into the host by the parasite. The eggs of the Dinocampus coccinelli are deposited by the female wasp into the parasite and into the body of a lady beetle, where the parasitoid larvae undergoes development. Then approximately 20 days later, the single prepupa emerges from the lady beetle's body and proceeds to spin a cocoon between its legs, which and it stays still. It doesn't move, and it causes it to show tremors, which is a defense mechanism against potential threats. And the ladybug remains alive and positioned atop the parasitoid cocoon, serving as a protective bodyguard. And approximately 1 week later, the adult parasitoid emerges from the cocoon. And it is notable to say that the lady beetle survives. It does not die. And the changes are likely to result from DCPV replication in the cerebellar ganglia. The Dinocampus coccinelli paralysis virus adds a third participant to the dynamics, which helps with the overall taking over of the host to provide nutrition and protection. So what does this mean? Overall, all susceptible organisms, mice infected with Toxoplasma gondii, grasshoppers from Spinochordodus tellini, and ladybugs with Dinocampus coccinelli, show that changes in locomotion or abnormal behavior that ultimately benefits the parasite in terms of reproductive success and survival is the ultimate goal. And even with different mechanisms and somewhat different results, the outcome remains the same: transportation of parasites to environments where they can grow and reproduce. Looking ahead, this field presents both exciting opportunities and formidable challenges, as there's more to the left to uncover compared to what we already know. And one potential avenue for future research lies in unraveling the precise molecular mechanisms that underlie the parasite-host interaction. And understanding this could help us deepen our knowledge of host-parasite dynamics and might unveil novel therapeutic targets. Thank you.
Thank you. I can confidently say I'm going to have nightmares tonight, so thanks for that. A couple questions coming in from the audience. First one, what initially drew you to the topic of zombie parasites, and what was the most surprising discovery you made during your research?
One of the things that drew me into zombie parasites was zombie movies. I really liked those as a kid, and learning that there's an actual thing called that affects parasites and makes them into literal zombies definitely made me interested in this topic.
Zombie movies as kids. I'm going to have a chat with your parents. Second question, how do you envision future research in this area evolving, and what specific aspects of zombie parasites do you think warrant further investigation?
Future aspects of this, it would definitely help with our therapeutic knowledge, because zombie parasites could potentially learning how they alter brain behavior, or like behavior through brain, could help us understand how our brain essentially works and what we can do to change anxiety levels and fear levels in people with PTSD, for example. Because as seen in the Toxoplasma gondii, mice infected with T. gondii didn't show fear towards cat urine and were actually attracted to it.
Got it. Thank you. Let me double check. I think that's all the questions we have. So thank you very much for sharing your work.
Thank you.